Alright, so focusing on mitosis, the division of the cytosol, if you look at a cell under a microscope during interphase, you're going to see that in the nucleus, which should be very clear, there's not really going to be a distinct chromosome quantity. You're not going to be able to see much there. It's going to be very spread out. I like to think about it in terms of like spaghetti. So like these chromatin molecules really are like you know, the DNA in its string form. Okay, it's not tightly wound, it's not bound up. It'd be really sloppy to try to split this up into two equal portions. So one of the first things we're going to have to do when we move into mitosis is to organize the chromosomes, to form our chromosomes and make them easier to divide. Okay, so the first stage of mitosis, and an easy way to remember the stages, as I mentioned to you in class, is that little mat that some people have in their house there for, you know, if they're out for a long period of time and they have a little dog, you know, has like little grass on it, and you know, a little container to capture the urine, but that's a PP mat, right? So prophase, prometaphase, metaphase, anaphase, telophase, PPMAT. Okay, so I hope that makes sense to everybody. And we're going to go through each phase individually. So in prophase here, the first thing that I want to make known is that the nuclear envelope is starting to disappear, it's starting to degrade. Also, we see the formation of chromosomes. Those chromosomes are still contained in that nuclear region. We also can see the start of centrioles. Okay, the next phase, which is called prometaphase, these guys start to move out of what that nuclear area would be. They're still not arranged in any kind of linear form or orientation, but they're starting to move. And this centriole structure with these spindle fibers starting to get enlarged. This is called an aster because it reminds early scientists of a star, and it kind of spreads out. Eventually these spindle fibers are going to attach to the chromosomes and kind of get them into their specific positioning and orientation, provide them like kind of a track to move on. And this phase again is called prometaphase. At this point the nuclear envelope should be completely dissolved, shouldn't be present, and our chromosomes should be very clear, and again we're moving them toward the area, the center of the cell. In this case, you guys, the other thing I want to point out here is that we have a total of 1, 2, 3, 4, 5 chromosome pairs. So actually this represents 10 chromosomes. Okay, and so what we're looking at is we have a chromosome and a sister chromatid, a chromosome and a sister chromatid. Okay, so we line those 5 chromosomes up in the middle. At this point, the spindle fibers are attached. They're lined up at an equatorial region. This is called metaphase. Meta means middle. Okay, and so this right here is our metaphase plate. Our centrioles are on either side in an animal cell. A plant cell does not have these centrioles. Instead, the spindle fiber will be anchored to the cell wall. Okay, but the chromosomes are lined up in the middle. They start to split out in anaphase. The centrioles are kind of cranking these spindle fibers in, and now we have 5 chromosomes that are going to go into this cell, 5 chromosomes that are going to go into this cell, and I hope that makes sense. Okay, all right, so here is our last phase. So in anaphase we have the cells, chromosomes are starting to split into 2 distinct regions. Okay, so the sister chromatids are separated now, and we have 5 chromosomes going to this cell here on the left, 5 chromosomes going to the cell on the right, and the centrioles are kind of cranking in on these spindle fibers and pulling those chromosomes toward either pole. In telophase, we're going to have those chromosomes are going to be polar. They're going to be found on either side, and you're going to see this thing called a cleavage furrow on a plant. So, in telophase, you're going to have these chromosomes move toward either pole, and you're going to see the nuclear envelope start to reform around them. In animal cells, you'll also see this cleavage furrow that will start to form. Okay, and so the splitting of the cytoplasm, which is not really considered part of mitosis, really the last phase of cell division, it is the last step here. This will continue to pinch in, and eventually will split the cytoplasm in half. Okay, so once the nuclear envelope is reformed, the chromosomes are going to unravel to make transcription and translation more accessible, and we'll have a finalized cell division. Okay, so at the end of telophase, we have cytokinesis, and then we'll have 2 distinct daughter cells. The last thing I just want to mention to you guys in this lecture or talk, or however you want to look at it, is some of our cancer terms. Okay, so just to get into a little bit more detail about this, what cancer is that the cells are not following normal cell division practices. That is, that adjacent cells are signaling to them and they should not be dividing, but they are. Okay, and once they start acting irrationally, I guess would be a term to describe it, what they'll do is they'll start to convert adjacent cells. So a cancer's growth can actually go through and through signaling really grow with quite a lot of speed and ferocity within a body. So there's a couple terms here that are important to understand and make note of. I have them listed here. I'll just go through each one. Okay, so a proto-oncogene is a gene that you would have from birth. Genes, remember, are a section of DNA that code for a polypeptide. Okay, and proto-oncogenes, typically everybody has a specific number of them that they would inherit, and what these are precursor to cancer genes. Okay, what a cancer's gene is called an oncogene, and those oncogenes, they can accumulate in one's body throughout one's lifetime. You also could be born with oncogenes and have cells that have cancerous instructions from early on. Okay, but typically what happens is they start as proto-oncogenes, and through mutation over the course of one's life, those proto-oncogenes will become oncogenes. Okay. In addition, you guys, we have genes that are utilized by cells to combat cancer's growth. Believe it or not, like, there are many instances typically within a human's life cycle where they would have cells that would start behaving in a cancerous way, and these tumor suppressor genes, what they do is they kind of step in and they would tell any kind of growth or cellular division that's irrational to kill itself, die, to go through apoptosis. And this is our first line of defense against cancer, and you know, the numbers on this obviously are not well known, but I would guess it would be very high throughout an individual's life, where cells would have mutations and then be recycled before they become an issue. Okay, and these tumor suppressor genes, again, are meant to suppress tumor growth. Not all types of tumors are dangerous. I hope everybody understands the nomenclature here, but a benign tumor is one that is not actively spreading or invading other tissues, whereas a malignant tumor is a tumor that is going to be invading. Malignant means fire, and it spreads very quickly if left unabated, and cancer screenings and things like this are so important to catch early and do something about them as quickly as possible. Okay, the last thing I want to point out here is the term mutagens. That's any chemical that is going to enable mutation of DNA. Okay, and just because you have a mutation, I don't want everybody to think of this as being like, you know, a negative thing. Mutations have led to some of the greatest advancements in organisms over time. Can be very positive, but for the most part, when we're talking about cancer or things like this, when we have alterations to our DNA, we have the potential to turn proto-oncogenes into oncogenes. We also have the ability to turn normal tumor suppressor genes that are functioning beautifully and make them inactive because of mutations within that genetic code. Okay, so typically what's going to happen is that we have pretty much a genetic aspect to our propensity for cancer, and that varies quite a bit from human to human. And then we have environment, which is the only one that we have control over. So really, you know, we can choose to live clean and eat healthy and exercise and do these things that have all been proven to reduce cancer rates, but that still doesn't prevent all mutation. Okay, so it's important to note that with increased exposure, the rate of mutation obviously would be increased. So if you choose to smoke, drink, you know, do these things on a regular basis, the rate of mutation would be increased. Okay, also as an organism lives throughout its life, the number of divisions increases exponentially, and therefore the rate of mutation is going to increase as well. So cancers typically tend to set in an older aspect of our population. They have a higher percentage chance of cancers across the board, and that's just because the rate of mutation increases with them. Most of the time cancer is not caused by one of these things, it's a combination. So you might have a mutation that caused an oncogene, and for the most part these guys have been active at eliminating any problem caused by the oncogene, but eventually a mutation occurs to these guys, so now you don't have that line of defense, and now a tumor starts to grow, and that has the ability, the potential to become cancerous. So I hope that makes sense. If not, please ask questions. Thank you. Take it easy.